Okay, so I'm really happy to be here. Thanks for giving me the opportunity to present today. And just a little bit about me. So I'm an assistant professor at Brigham Young University. So this is an aerial shot just to kind of give you a feel for where I am. It sits at the base of the Wasatch Mountain Range, which is basically next to the Rocky Mountains. And we're here at BYU. We're really proud of our undergraduate program in bioinformatics. We have probably 70 or 80 students right now for undergraduates, and if any of you are looking for graduate students, I'd be happy to talk to you about possibilities with that. Okay, and before I start, I just want to give some acknowledgement. So the work that I'm going to present today is collaborative work with various different people involved, and particularly I want to mention Andrea Bill and Evan Johnson, who are my colleagues at the University of Utah and Boston University School of Medicine, and Shelley McNeil and Nader Elchar. They've been graduate students who have done a lot of the work on these projects, so I definitely want to acknowledge them. So to start out my talk, I want to just give some brief background about cancer and pathways. And so one of the well-known examples related to this concept is from colorectal cancer. Back in the 80s, people started to discover more and more that it wasn't just one single gene or one particular mutation that leads to colorectal tumors, but rather that it was a series of events that occur at the molecular level over the course of time, sometimes decades, that enable normal colonic cells to transform into adenomas and eventually into carcinomas. And so the idea here really is that it's not just one gene, but multiple genes that may need to work together in order to cause such transformations. And more recently, so this is from 2007, when it was people were first able to look at mutations across thousands of genes, it was found that there were lots of mutations occurring within tumors. There were some genes, so you can kind of see the peaks there on this diagram, that what they call mountains, or represent genes that are mutated relatively frequently. They had some hills that are mutated somewhat frequently, and then a long tail of mutations that occur in a lot of different genes that occur very infrequently. And so part of the challenge is to take into account not just the really frequent mutations or other types of events that occur, but to account for all the various types of events that happen. So one thing, this is from the same paper, one way they did this was with a simple pathway analysis, where if they look just at PIK3CA, which is a gene that's relatively frequently mutated in breast cancer, so now we're shifting to breast cancer, they found that one gene was mutated in about 11% of the tumors that they profiled, whereas if you take into context the whole pathway, some type of aberration within that pathway was observed in about 33% of those tumors. So by placing it in that biological context, they were able to learn more about what goes on within breast tumors. Even more recently, this is a paper from the Cancer Genome Atlas. So they found a similar thing, that multiple genes and multiple types of events were playing a role, but it wasn't just somatic DNA mutations, but other single point mutations and various types of aberrations that can occur. And so the challenge really is to take into account all these different types of aberrations, all these various types of genes, and understand more about the biology that's going on within these tumors. And this is how pathway-based approaches can be useful. There are a lot of curated pathway databases that are out there. For example, KEGG, BioCarta, Reactome. These represent current existing knowledge about what pathways play a role in various different biological processes. So we have these curated pathway databases, and the challenge is to take those and map that type of curated data to the actual gene expression data or other types of data that we can get to profile tumors with. This is just an example of one pathway within the KEGG database, and that represents the proteins and genes involved in cell cycle regulation, Some of these pathways are very large and complex, whereas others are more focused and very specific. So today I'm going to talk about two different pathway-based analysis methods, or ways of accounting for this complexity and mapping those curated databases, or in one case mapping those curated databases to actual tumor data. One of these approaches I'll call gene set-based approaches. The other I'll call a signature-based approach. And I'm going to focus initially on gene set-based approaches, which the general goal of these is to identify gene sets that show consistent differences at the molecular level between biological samples that represent two conditions. So maybe these are samples where we know that those are tumors that responded to a drug, and you would compare those against tumors that did not respond to the drug, and your goal would be to identify pathways or processes that are different between those two types of samples. Alternatively, with signature-based approaches, we can also use that to identify, one way to use that is to identify pathway activation status in individual biological samples. And I'll get more into detail on what I mean by that later on in the talk. So typically gene set-based approaches are applied to RNA expression data. The most well-known methods are applied to that type of data. However, more and more methods are being designed to be applied to other types of molecular data, including DNA variation, protein expression, epigenetic variation. And probably the most well-known of these methods is called gene set enrichment analysis. This has been around for over a decade now. Just briefly to give an overview of how this works. Say you profile two different types of samples, so maybe samples that responded to a drug and samples that didn't. So those would be class A and class B on the left side. Say you had gene expression values for those samples. What you could do is that what you would do with this method is you would, so each row here represents a gene. What you would do is you would take those values or those genes and rank them according to how different the average expression value was across those two different types of samples. So in this case, the genes at the top are the genes that are overexpressed in group A relative to group B, and the genes at the bottom of that heat map are the genes that are overexpressed in group B relative to group A. Once you've done that, you take a gene set or a set of genes that you know participate in a certain type of process, and you see where those genes fall in this ranked list. And what you would do then is look to see, are there gene sets where the genes are clustered toward the top or the bottom. So if they're clustered towards the top, as you see here in this example, you could conclude that those genes, or overall that gene set is upregulated in those samples, in group A, for example. And you do that for all the gene sets, and then you apply some statistics to try to understand, okay, which gene sets are most likely to be playing a role in this phenomenon of interest. This has been a really popular method. As you can see here, it's been cited thousands of times, and it is a really nice method, though there are some limitations of this. For example, it applies statistical tests to individual genes, or one gene at a time, and thus it doesn't explicitly account for interdependencies among genes, which we know do occur quite frequently. We know there's strong correlation often between different genes when you're looking at gene expression data. Another limitation is that this method is looking for gene sets that are either upregulated as a group or downregulated as a group, not necessarily gene sets where some genes might be strongly upregulated and other genes might be strongly downregulated. Another limitation is that this method is primarily designed for gene expression microarray data, whereas we'd like to be able to apply this type of an approach to many different types of omic data. Various other methods have been out there. Been put out there. One that I really like is by Peng et al. This was published in 2006. They use the random forest classification algorithm. So this is a machine learning algorithm, and the benefits that you get from using a machine learning or a classification algorithm is that such algorithms can account for interdependencies among genes. They're designed to identify patterns that are different between sets of samples, and tend to be really good at doing that. It can account for up and down regulated genes simultaneously, and they're intended for general application. So these types of algorithms are applied in various different scenarios in the business world and in biology and various other areas. And so their idea was, could they take those algorithms and could they overcome some of those limitations of existing methods? Just to give just a really simple example of how a classification algorithm works. So let's say you had a bunch of produce that you bought at the supermarket. You wanted to develop a classification model to distinguish between fruits and vegetables, so you could get all of those produce items. You could identify various characteristics about those objects, and for example, their color, their shape, their size, their smell, whether they have seeds, all types of things like that. And then you could use a classification algorithm to say, okay, based on these attributes that we observe in these items, where we know whether something is a fruit or a vegetable, let's train a model, and then now let's apply that model to some other type of produce item where maybe we don't know what that is. And so maybe we could use that type of algorithm to answer the age-old question of whether a tomato is a fruit or a vegetable, for example. So just to give just a simple example, in the way Ping et al. Used it is they applied this to data sets where, say, you had a gene expression data set where you have maybe 20,000 genes, and you have data for maybe 100 patients. And so the random forest algorithm in this case is going to look for patterns that are different between the two different types of samples, and then make predictions and see how well it can predict and identify which group each sample belongs to, and they use a method called cross-validation for that. There's various types of machine learning algorithms. This is just a simple diagram of the random forest algorithm. It's based on the decision trees methodology, where you're looking for ways to differentiate between the two groups using the various attributes within the data, and then you apply that multiple time with random perturbations to the data, and you look and see, okay, what's the consistent message that we're observing in the data. Another type of algorithm that's used commonly in the bioinformatics community is the support vector machines algorithm. So the way this works, this example here is just a really simple example. Suppose you had gene expression values for two genes, gene X and gene Y. If you look at the data on the graph, you can see that there's a clear difference in the expression patterns between those samples that had cancer and those that were normal, but there wouldn't be a way to draw just a straight line through that data and distinguish between cancers and normals. So the support vector machines algorithm, what it does is transforms that data to a higher dimension, and it can get really complex depending on how many genes you have in your data set, but it looks for a decision surface or a way in that higher dimension to distinguish between those two, in this case, cancers and normals. Let's see. Okay. So we really liked the Ping et al. Method, but there were some ways that we wanted to improve upon it and make it what we felt would be more useful. So we developed a tool based on a similar methodology, but had the ability to use many different types of algorithms. So for example, support vector machines made it so if you wanted to use that algorithm, you could use that, or if you wanted to use random forest, you could use that, or something else that you liked. We designed it so that it could be applied to multiple types of omic data, either separately or in combination, really under the mindset that looking at one particular type of data by itself will give you some information that's useful, but if you could apply it and learn from multiple types of data, that it may give you a better signal or be more informative than just looking at one type of data alone. A couple other little things we did with this is that we ranked the gene sets by area under the ROC curve rather than by classification accuracy, and I'll explain more in a minute how our methodology works and where that fits in. But the advantage that you get from this is that if you have a data set where you have a strong imbalance between samples in either group, say maybe 90% of the samples belong to one group and 10% of the samples belong to the other group, the area under the curve is a better metric for handling, or I guess measuring accuracy in those types of scenarios. And lastly, and I'll talk about this in a minute, but our method corrects for gene set size. So if you have a pathway or a gene set with a lot of genes, maybe it would be more likely to give you better accuracy than a small gene set, and so we attempt to take that into account. This is a schematic of our overall approach. So the inputs to the algorithm are different types of omic data, whatever you might have, and that could be, like I said, separately or in isolation. And you input that as well as a curated pathway database. And what we do with that data is we map the omic data onto those pathways. And so let's say for pathway A, we extract all of the gene expression values or DNA variation values or whatever it is for that specific pathway. Then we use cross-validation to see how accurately can we distinguish between the samples in either group, Class A or Class B, using just the data from that one pathway. By default, we use the SVM classifier just because that's an algorithm that I've had a lot of success with in the past. Like I said, there's the ability to try multiple different algorithms if you'd like to do that. What you get out of that is for each of those pathways, you'll get an area under the curve value. Higher values indicate that you have a better ability to distinguish between those two groups. I'll talk about the p-value in a second in FDR. So let's see. So yeah, let me show you this graph here. In our preliminary analysis, what we found was that there was indeed a strong bias toward larger gene sets. So you can see that on the left side, that gene sets with a relatively high number of genes tended to have much higher AUC values. There was a strong correlation there. So what we did is we used an approach to correct for that. So we let me go back to this slide. So if you look at number 2 there, instead of using these curated pathway or curated gene sets, we generated a random database of gene sets, or a bunch of random databases of gene sets. We repeated the cross-validation process to get a null distribution to see what kind of accuracy would we get just with randomly selected gene sets. And so the idea there is what level of accuracy do you get for a gene set of size X for the actual gene sets versus random gene sets. What we found, and then we calculate an empirical p-value based on that null distribution. What we found was that after doing that correction, our p-values showed basically no correlation between the size of the gene set and the metric that we were using to quantify how well it could distinguish between those groups. So the next thing we did was we wanted to test this on simulated data. So we generated a simulated data set that had multivariate patterns within the data set. So it wasn't just, generating generating values where one gene at a time might show differences between two groups, but where there were multivariate patterns that differed between those two groups, such as you might see in an actual biological data set. For that, once we generated those values, we then also created a gene set database that we could use to test this. And some of those gene sets in that database differed in size. They had anywhere from 25 up to 300 genes within each of those gene sets. And then within each gene set, there were anywhere from 0 to 50 what we call signal genes, or genes that in our simulation showed significant differences between either group in our simulation. And so what we found was that as long as there were at least 10, at least on average, as long as there were at least 10 signal genes within a given gene set, our method was able to identify that that was a gene set that was relevant or important. And the threshold we used initially was 0.2 for the false discovery rate. So we found that consistently, as long as there were at least 10 signal genes, that it fell below that threshold. Whereas if we apply the gene set enrichment analysis method, what we found was that there had to be at least 30 signal genes within a given gene set in order for that method to identify that gene set as being relevant in this analysis. So we applied a few other methods, and what we found was that our method for various different FDR thresholds was able to identify a higher percentage of gene sets that it should have identified based on our simulation compared to other methods. We also applied this to a data set that was simulated in a similar way, but had a 90-10 split. So some samples, there were 90% of samples that were in one class, 10% of samples that were in the other class. And what we saw was that looking across all of these methods, the performance of all of them went down quite a bit in this analysis. However, our method still tended to identify a higher proportion of gene sets that were simulated to be relevant, or that should have identified. This data here, this is from data based on data from the Cancer Genome Atlas, breast cancer data. So one of the nice things about TCGA, the Cancer Genome Atlas, is that it has multiple, for the same samples, it has gene expression data and other types of data that have been profiled using multiple technologies in some cases. So we looked at samples that had been profiled using gene expression microarrays and other samples that had, or the same samples where they had also been profiled with RNA-Seq. What we found was that if we applied GSOA to that method, and in this case we were specifically looking for differences between samples that had a HER2 amplification and those that didn't. What we found was that there was a really strong correlation between the results of our GSOA method, strong correlation between the two technologies for both the AUC values and for the p-values. So for the same type of comparison, comparing HER2 positive and HER2 negative breast cancers, we applied this to RNA-Seq data, but also copy number variation data from the tumors and somatic mutation data. We found that RNA-Seq gave us overall the highest accuracy levels. CNV did pretty well, but somatic mutation data didn't do quite so well. What we concluded after looking at, or our thought was after looking at this was, although somatic mutation data, or although the accuracy varies for these different types of data, potentially if we combine all the data together and just let the machine learning algorithm try to pick apart or identify what is the most relevant signal in the data, maybe we could still use that somatic mutation data, have it be useful, and tell us something about the biological processes that are going on within these tumors. What we found when applying that was, this is just showing the top 8 pathways that we found. The ones that are bolded are the ones that these are ones that we kind of would have expected to see, or they're pathways that are reflecting activity that plays a role within cancer in general, and then also you would reasonably expect to see come to the top of your list when comparing samples that had a HER2 amplification or not, pathways related to growth factor signaling and such types of things. So, we can analyze the data on the computer, but really what we hoped to find was some way of validating experimentally that this approach was giving us something meaningful or biologically relevant. So we took this method and applied it in a different, to a different type of problem, so trying to identify biological processes that influence breast cancer risk. So we had data for individuals who had a strong family history of breast cancer. They had at least 2 first-degree relatives who had developed breast cancer, and some of these individuals had developed breast cancer by the age at which you might expect they would, whereas others had not developed breast cancer by a similar age. So what we wanted to see was, can we identify or understand more about what are the processes that are playing a role in determining whether someone develops hereditary breast cancer or doesn't. And our patient cohort included individuals who had some individuals who had BRCA1 or BRCA2 mutations, which we know play a role in breast cancer risk. Then there are also individuals who we call BRCAX individuals who did not have a known mutation in these genes. So we were trying to identify biological processes that are complementary to or alternative ways of influencing whether somebody develops hereditary breast cancer. What we consistently found in doing this analysis was that pathways related to cell adhesion processes came to the top of our list. We tried various different, or we applied it to data from 2 different populations, and we consistently found that pathways related to cell adhesion came to the top of our list. So then we looked to see, could we use experimental methods to try to gain additional confidence that our in silico approach was giving us valid results. And also looking at the literature and getting a sense that altered cell adhesion is a reasonable it makes reasonable sense that cell adhesion could play a role in breast cancer risk. We know that as the adhesion processes between cells are disrupted, that can cause cells to misbehave, it can cause cells to act in a more invasive manner and can cause cells to become malignant. So 2 ways that we used to, 2 assays that we used to validate this. We took cells from women who had a family history of breast cancer and from controls. In a dish, we simulated the extracellular matrix environment. We put the cells in this environment for either of the conditions. What we found was that cells from women who had a family history of breast cancer tended to adhere to the simulated extracellular matrix less well than cells from women who did not have a family history of breast cancer, suggesting there was something fundamentally different about those cells between those 2 groups. And another thing we did was we used fluorescence microscopy to look at these cells from these 2 different populations, individuals that had a family history of breast cancer and those who didn't. This is just sort of some of our, I guess I picked a couple exemplary sets of cells to look at, but what we found was that, at least for this presentation, what we found was that in the control cells, or those who didn't have a family history of breast cancer, the cells have a very different structure under the microscope. So they tend to be more tightly connected to each other. They tended to just have a different structure, whereas the high-risk patients tended to be more spread out, not as highly connected to each other. And again, we use this as, this gave us additional confidence that the in silico result we had obtained actually does point to processes that occur within cells that may influence whether somebody develops breast cancer or not. Okay, so our method is available as an open source software package. You can access it from my website or on Bitbucket. And we just barely got a paper published, or had a paper published on this in the last few days. And we'd love to have you read it and give us any feedback you may have on that. We want to go a couple directions with extending the method. So, for example, using ensemble-based approaches. Maybe for one type of omic data, random forest might be a good algorithm, whereas for another type of data, maybe k-nearest neighbors or some other type of classification algorithm is a better one for that type of data. And so one way to approach this is to apply various different algorithms to the data, and then there are ways of combining or aggregating evidence across those different algorithms and make it an aggregate prediction from that. There's also a challenge with redundancy among pathways, so we have some ideas on looking into that and trying to make it so that the results that you get from our method are more easy to interpret or act upon. And then lastly, I would like to support multi-class problems or cases where maybe you don't have two groups, you don't only have two groups of biological samples, but maybe you have 3 or 5 or 50 different groups, and you want to be able to identify patterns that are consistently different across those groups. Okay, so now I'll shift gears to signature-based methods. So just as I mentioned earlier, the goal of these methods is to, at least the way we're using it, is to identify pathway activation status in individual samples. So identify whether a given pathway is turned on in a particular biological sample or tumor sample. So the way these approaches work is kind of similar to links, is that you have cell lines where you treat the cell lines either with, or you perturb the cell lines in some way. So in this case what we're doing is we're perturbing the cell lines by activating a particular pathway or a particular gene to see, and then we measure gene expression levels for those cell lines and for controls to see what are the differences in behavior in these cells when that pathway is activated or not. So I use the term pathway a little bit loosely in this context. One example that I'll talk about in a minute is, say what we would do is we would activate the RAS oncogene within the samples, and we would see, okay, when RAS is turned on, what is the effect on expression levels within those cells? And so then we infer from that type of effect is the downstream effect of RAS being turned on, or that's the activity that's associated with the RAS pathway. Over here on the right is our signature, so you'll have some genes that are turned off when it's activated or turned on when it's activated. We take that signature and then we apply that to, for example, to a tumor sample to see, okay, based on the patterns that we observed in vitro, which tumors appear to have patterns that would correspond to that, or that would indicate that that pathway is active within those tumor samples. We looked at the RAS pathway for one reason, because it's considered to be an undruggable target. So we wanted to see if there were alternative ways of targeting RAS without necessarily trying to target that particular protein, but to target the pathway as a whole. And so the question we asked was, for which drug repurposing candidates do we observe a strong correlation between RAS pathway activity and drug IC50 values? So first we took our signature that we had generated using the cell lines initially, the initial perturbation, and we applied that to a separate set of cancer cell lines. There are about 35 of those cell lines. And then we looked to see, okay, in which of these 35 cell lines is our approach indicating that the RAS pathway is active in those cell lines. And then we also used a dose response assay and applied that to the same panel of cell lines to see in which cell lines is a particular drug or particular combination of drugs, in which of those does it appear to be most effective. So then what we did was look at a correlation between those two. So on the x-axis here is the predicted signature-based probability of RAS pathway activation. On the y-axis is EC50 value, EC50 value for those cell lines. We saw, this is just showing the one result that we really focused on in this analysis, was that for a combination of Gefitinib and then a drug we're calling U0126, for that combination of drugs, there tended to be a relatively high correlation between RAS pathway activity and the EC50 values for those cell lines. One thing that was interesting that came out of this was that there wasn't really a strong correlation between KRAS mutation status in the cell lines and the EC50 values. So it appeared that you couldn't just rely on the mutation status in order to indicate whether individuals would respond to this drug combination, but that you needed to take into account the pathway as a whole in order to account for that. We're looking into this further, and I don't have time to go into all the details, but looking into this further using Western blots and other experimental methods, what we found was that Gefitinib, which we know it's designed to target EGFR, so it was targeting that component of the pathway. It was kind of upstream of KRAS, but then U0126 was targeting the MEK protein, which is downstream of RAS. So this led us to the conclusion that, rather than targeting RAS itself specifically, perhaps this approach was effective because it targeted, this combination of drugs targeted multiple components of that pathway, and accordingly was more effective for doing that. Just a couple notes. So I was thinking, how does this relate to links? So we know that in links there are lots of gene activation signatures where a particular gene is turned on or turned off. And so you could use a similar approach where you could look at the gene expression values for those different conditions. You could apply that signature to tumor expression data, maybe from TCGA. That would give you, for each of those tumors, it would give you a predicted pathway activity. And then you could use a similar approach using some of the drug treatment perturbations and get predicted responses for the same tumors. And then you could look and see, okay, for which drugs and which pathways do we see a strong correlation for these tumors between pathway activity and predicted drug response, and then you could take it from there and try to learn more about what's going on, or what's the relationship between that pathway and that drug, or potentially combinations of drugs. This is my contact information. I'd love to hear from you, and I'm happy to answer any questions.
Okay, this is Jimba from FDA. I have a question for you. You mentioned that you use pathway, and that is a very nice approach, and by the way, it's a very nice presentation. My question is that, so you have cancer patient sample and you look at their genes, and you were talking about linking gene signature to pathway. Now my question is that you have many gene mutations. How should we, are the pathway that you identify, would they be related to the so-called driver mutations? I'm trying to learn in terms of driver mutation versus the pathway that people pick up in terms of response to drugs. Driver mutation, when I say driver mutation, I'm learning that actually patient could become resistant to a drug after treatment, and they have so-called mutation, a driver mutation that cause drug resistance to a certain drug treatment. Right. So what is the benefit of pathway analysis versus trying to understand the driver mutations?
Yeah, thank you. Let me try to answer that by going back a few slides. So going back to this one here, well, this one here actually. So in this case, we for these cell lines, we looked at KRAS mutation status. So in this case, you could think of the KRAS mutations as drivers, potential drivers of those cancers. But we found, at least for this combination of drugs, that there wasn't really a difference in response based on that mutation status. Whereas if we looked, instead of looking at the mutation status, if we looked instead at the downstream gene expression values for those cell lines or for that pathway, we were able to see a strong correlation. And so suggesting that we may not know all of the underlying mechanisms that drive how tumors develop and how they behave. So instead of, or maybe not instead of, but as an alternative to, or in addition to looking at those underlying mutations and trying to identify what are drivers and what are not, we can potentially look at the downstream effects and use those as a way to guide treatment and maybe to overcome treatment resistance in that way by targeting, not just one particular gene, but targeting the pathway in general.
Then I have another question for you. So NIH recently just initiated the Exceptional Responder Initiative. So clinically, we see some of the patients respond to a drug that was not approved for treating that particular cancer. But somehow the cancer, I'm sure you heard about this, and the patient, that particular patient responds to that drug.
Yeah.
And from last week, their presentation at the FDA was that they have 75 patients and their tumor sample, and the cancer, they cover a wide range of cancer. My question is there a way that we can use computation, but instead of using statistics trying to see the significance, is there a way that we can do in vitro subgroup analysis based on the tumor sample and to identify early on who will be response to a certain treatment exceptionally well?
Yeah. Let's see. I mean, I think in this case that you would be comparing exceptional responders against people who were not exceptional responders or regular responders. And so, there are different things you could do. You could, using the first, gene set based approach, you could use that to try to identify gene sets that are biological processes that are behaving differently between people who respond to the drug or not, and that may help you to understand more about why those people are responding or not responding to that particular drug based on the mechanism of action of that drug or that kind of thing. That's one answer. Another answer would be using a signature-based approach. One approach would be to look for pathways that are turned on or turned off in exceptional responders in individual patients, and then use that information to try to guide you in determining what treatments to use or which ones will be most effective for those patients.
Thank you. I'm wondering whether pathway analysis will be too high level and that we may miss some patient or some specific gene mutations.
I'll just briefly answer that. So I think, it's a legitimate concern that if you're looking at the pathway level, it is relatively high level information that you're getting out of it. In some cases that may be useful, like we feel like in this case with this drug combination that I showed here, that it's useful even though we may not understand all the underlying mechanisms as to how these drugs target these tumors effectively. It still does provide useful information for us. However, if you're trying to get in and pick apart the exact mechanisms or get in and dig into more detail, there would definitely be some additional steps that you would need to do in order to dig down into that and understand more.
Thank you.
Thank you. Any other questions?
I was wondering if you can go over the next slide.
Yeah.
I was wondering if you can go over the next slide, not this one, the one that you measured, the correlation one.
Yeah.
This one. Just if you can explain what was measured again, and then how you can potentially improve this correlation, because it still looked pretty random, even though you got a nice Pearson R. I wonder what could be done to make it better. But first, if you can just explain it a little bit more. like what specifically, I think, what was measured? Like, was it RAS? When you said RAS pathway activation, what was exactly measured?
Okay, let me back up a couple slides with this. So let's see, right here. What we did was we activated RAS within a cell line, and then we measured the gene expression values when RAS was active versus when RAS was not. That gave us our signature that indicated, okay, which genes are turned on or turned off when RAS is active within those cells. And then we used that data to make a probabilistic prediction for each of these cell lines that I'm showing here. Basically, for a panel of 35 cell lines, we used a machine learning / statistical approach to generate a probabilistic value for each of those cell lines that indicated, okay, based on this signature here, what is the probability that that pathway is active within those cells? So that's what you see on the x-axis here, is those probabilistic values, one for each of those cell lines.
How did you activate RAS, like in that experiment? Is it overexpression?
Adenovirus-based overexpression.
Okay, thank you.
Yeah. And then to answer your other question, as far as getting, refining these results, one way to do that is to expand this to additional cell lines. Maybe look at protein expression levels and generate a signature based on those, or maybe even look at other, other variations on these drugs that maybe have a more are more effective in combination with these cell lines. Those are the ideas that come to mind, but I'm certainly happy to hear other ideas that people might have.
I think the idea of overexpression and then looking at the signature of overexpressing some of those other pathway components, or in general, like if you look at a database of, if you're just trying to make it maybe in large scale, looking at overexpression or down regulation of many, signatures, and then you try to correlate those signatures. Like maybe you can collect those from like GEO or something, and then if you try to correlate your sensitivity response with the correlation of the gene expression signatures from the public domain, you may be able to find some signatures that you have even more, like higher correlation.
Right.
And those can be potential indicators of the pathways that are resistant versus not resistant. But overall, I think it's a really interesting, nice approach.
Okay, well, thank you very much, everybody, for participating. I appreciate it.